You're listening to the Future Tech Podcast with Richard Jacobs. Future technologies such as artificial intelligence, stem cells, 3D printing, gene editing, Bitcoin, blockchain, the microbiome, quantum computing, virtual reality, and exploring space are much closer than you might think. In fact, many early versions of these technologies are in play right now, and the companies that are using these technologies are the focus of this podcast. My goal for you, the listener, is to learn from these podcasts. You may very well learn something that may change the course of your life for the better, steer you towards a new career, or give you insight into addressing a thorny medical problem. Remember, this podcast and its content is informational in nature only. No medical, tax, legal, financial, or psychological advice is being given. If you've enjoyed the podcast, please listen, subscribe, like, and tell your friends about it. Thank you. Hello, this is Richard Jacobs with the Future Tech and Future Tech Health podcast. I have Guru Banavar, Viome's Chief Technology Officer, and the company is Viome. It's like Biome, but with a V. So, Guru, thanks for coming.
You bet, Richard.
Yeah. Well, first of all, Viome itself. I know we've spoken to two people at Viome before, but in your own words, what's the mission of Viome? What are you guys about?
Viome is about eliminating chronic illness. And we think that the way to do that is to first understand the gene expression within your biology and use the best available food and supplements to help get your gene expression to a healthy, long-term, preventative kind of a state for you as an individual, meaning personalized, as opposed to just doing a one-size-fits-all for everybody. Rich, does that make sense? Can you hear me?
Yeah, no, that makes total sense. I was going to joke and say, food and supplements are a lot better than trying to knock out my Krispy Kreme gene, to prevent me from eating that. So much more natural way.
Yeah, we think doing this in a natural way, going back to sort of the basics and getting totally connected with the right kinds of foods and maybe even turning into some of those foods into supplements and food extracts, including all the changes that that are required for your most interesting and important organ, which is the gut microbiome. That's what we think we should be doing. And the reason we picked, of course, the gut microbiome is probably pretty clear to many of your listeners that the gut microbiome is the most important and known as the second brain. It's the most important organ that drives many of the chronic illnesses that we've been seeing especially in the developed world in the last few decades. There's been a tremendous increase in the number of people who have chronic illness. Pretty much everybody around you, if you look around, every other person probably has some kind of chronic illness, whether it is some kind of an autoimmune disease or some kind of metabolic or neurological or, one of those many different types of things, including cancer, right? All of these are chronic diseases that everybody suffers from. And we think it's because sort of the pendulum swung too far in the Industrial Revolution where, we started kind of going after the microbes, all microbes, even though we meant to only handle infectious diseases. We literally, had antibiotics and we had all kinds of antimicrobial, liquids and sprays and what have you everywhere around us. And so we basically demolished the microbiome inside our own bellies. And that has actually caused a tremendous imbalance in the microbiome in the last few decades. And we've been discovering that, having and restoring the right kind of activity, and that's the key word, activity, in your microbiome will and, probably is the most reliable way to get people back into optimal health.
Well, the difficulty is, from what I've read and seen, everyone's microbiome is different. No one's really sure how different. But how do you know what someone's healthy state was versus their current dysbiosis? And
how do you restore them to a healthy state? How do you figure that out?
Yeah, so the way to figure it out is through a lot of data, right? So what we do, and this is my job in Biome. So I am very, intimately familiar with all of the data we get. Plus, I'm also very passionate about how to use data to understand what goes on in very complex, large systems like the microbiome. So the way we do it is, first of all, we collect data from thousands and thousands of people. At this point in time, Viome is getting close to 100,000 customers, and so that's a lot of data, right? So when you think about that many samples, each sample, stool sample from a customer has approximately, a few thousand, let's say 2,500 or so different organisms that we can see being active. Mm-hmm. We can see somewhere around, 8,000 or so genes, or, actually these are not individual genes. These are sort of collections of genes that may come from different organisms. So these are clusters. So we see 8,000 or so clusters of such genes that are active in the microbiome. And those kind of represent functions that are happening in your microbiome, such as, for example, the production of certain metabolites can be considered a function. And that doesn't happen just with, an organism, but it happens because there are genes that are active across multiple organisms, and they together make certain metabolites. I mean, including vitamins, for example, your vitamin K, for example, that is generated in your microbiome, that's a function. And we can see these functional groups of activities in your microbiome, in somebody else's microbiome, and so forth. But when you have so much data, we can see what is the difference in these patterns of activity between people who have a particular disease like irritable bowel syndrome and people who don't have that disease, right? So we can see what the differences are between these populations, and those differences tell us how to restore, the patterns in the people who have IBS to the patterns potentially, right, of the people who don't have IBS. That's how you tell the difference.
So what, I know, some things you can't say, but what can you say, for instance, in IBS or any other condition? What differences have you seen in looking at thousands of different microbiomes?
Yeah. So look, so the way I'll put it is, we are seeing differences in disease versus healthy populations one by one. Okay, so let me start with glucose response, right, which is actually fundamental to diabetes. So we just wrote a paper, and the paper is available on bioRxiv, and that tells us that the difference between people who have good glycemic response, meaning that they don't have a very high glucose response even for foods that may have high levels of carbs, right, have certain pathways in their microbiome that are more active. So, for example, there's a pathway called the indole acetate pathway. That particular pathway is, it consists of a number of enzymes and genes that are all acting together to actually, contain the glucose response of individuals, or, that is driven through your microbiome, basically.
Right.
So we see that as one of the examples. On the other hand, if we see that, there's another pathway called the fucose pathway that is positively correlated with your glucose response, meaning that if that pathway is more active, then we see that when you eat a food containing more carbs, your glycemic response, your glucose response will be much higher. So there's a whole list of such pathways that we see as the differences between people who have high glucose response versus people who don't have high glucose response. And that's published in this paper that I just mentioned. So that's the first thing that we've already published. Now, it turns out for irritable bowel syndrome, there's again a series of these pathways that are going to be more active for people who have IBS versus people who don't. Now, because we have not yet published that paper yet, and we will be publishing it shortly, I cannot talk about it right now and give away all the answers before we publish it. But suffice it to say that we have seen that certain pathways are more active in people with IBS and more active in people without IBS, and those will be published very shortly.
Well, in terms of glucose response, what happens if you see someone with a microbiome where their glucose, their pathways are missing, whatever it is? So the glucose uptake, I guess, is diminished. Does that mean that more undigested glucose, for instance, comes out in the feces? Like, what happens to it if someone has, let's say, they don't respond very positively, they don't respond very much to high carbohydrate foods. For some reason, their body just doesn't, produce a lot of the, convert it to a lot of sugar. What happens to it? Do you know?
Yeah. So look, carbohydrates are a food substrate that gets translated to many things. Okay, it's not just absorbed as glucose and processed by the human cells, but it's actually processed by your microbes to generate a number of different metabolites. So, for example, there is a whole class of metabolites called short chain fatty acids. And one example of a short chain fatty acid is butyrate. Another example is propionate. Now, these short chain fatty acids are generally very good for your inflammation, meaning they're anti-inflammatory in nature. So one of the things that the bacteria in your gut microbiome do is to translate certain kinds of carbohydrates into butyrate. And if those translations or conversions happen because your pathways for butyrate metabolism is functioning well and is functioning at a high level, that would be a great piece of news for you, because that will result in lower inflammation in your gut lining, which is considered to be one of the fundamental factors for chronic illness of many different types, right? So there's this whole idea of a leaky gut or an inflamed gut that could be causing a whole range of different issues in your body. And irritable bowel syndrome, is definitely one of those diseases that have been given a name after, various kinds of inflammation could be happening in your gut. And the more direct disease that people associate with inflammation, of course, is inflammatory bowel disease, right? So inflammatory bowel disease is of multiple kinds. One of them is, called Crohn's. Another one is called ulcerative colitis. Now, these are all happening because the anti-inflammatory properties of your gut are not very active. And in fact, the pro-inflammatory properties of your gut may be overly active, right? And there's both pro-inflammatory and anti-inflammatory activities that your microbiome does. And so one of the things that we can do in Viome is to see, out of all the pathways that are active in your microbiome, which of your anti-inflammatory pathways are active and which of your pro-inflammatory pathways are active, and give you essentially some scores. And if you look at your Viome app after you send your samples in, we give you scores for your inflammatory activity. It could be low, medium, or high, and you can tell whether is my inflammation high because all of my, pro-inflammatory pathways like LPS, for example, or anti-inflammatory pathways like butyrate, all of these are, in a certain combination, because these are all sort of combined together to give you your inflammatory score. And,
right.
and so what we end up doing is we use all of these pathway scores to determine what are the foods you should be eating so that you can turn on or turn off the appropriate pathways. So, if you see that there's some substrate that if you eat more of, you can increase the anti-inflammatory pathways, then we propose that recommend that for you as an individual. And the idea is that if you do follow those recommendations over time, you should be seeing an improvement in the inflammatory activity of your gut. So that's kind of the whole series of reasoning that we do in our right. AI system.
So is the appearance or lack of pathways, does that correlate downstream with which species will appear or won't appear in the gut in large amounts or small amounts? Meaning there's no food for subsequent species, so they're not present?
So, Rich, I wanna just state that looking at species or strains is not actually the best way to look at your microbiome. And, even though 99% of the literature in this field has been looking at taxa, meaning species and genera, genus, or strains, that's actually not a good way because, these species or these bacteria are just arbitrary collections of, genes, right? Each, bacteria typically have about 3,000 genes each, but it turns out that multiple bacteria can have the same genes, meaning that, bacteria A and bacteria B can both have anti-inflammatory genes or pro-inflammatory genes. So just because bacteria A is active doesn't mean that it always has an anti-inflammatory effect on you. Or bacteria B is active doesn't mean that it always has a pro-inflammatory effect on you. It can be the other way around, because both bacteria may have genes and pathways that have either pro-inflammatory or anti-inflammatory. So what is important is to look at the genes themselves and the activity of the genes themselves that underlies these organisms. And that's what we do at Biome. We look at all of your genes and we say which genes are active regardless of which organism it comes from. So when you start seeing which genes are active, we can say, oh, look, your anti-inflammatory pathways are high. We don't care which organisms it comes from, because those organisms are not a reliable way of saying they're always going to be anti-inflammatory, pro-inflammatory. So we simply say your anti-inflammatory pathways are doing well regardless of which organisms they come from. So one of my key messages today is that looking at your microbiome just as species or strains or genus level, that's not at all the right way to look at it. What you should be looking at is what are the functions that your microbiome is active for. And that word active is my second message, right? It's not just about what DNA your microbiome has, but it is which DNA has been expressed as RNA. And that means, it's active, as opposed to all the potential DNA that could have been active. We actually see what is the actual RNA that is active, independent of which organism it came from. And that's kind of how we compute all of these scores that we show you in your Viome app, if you get it.
Yeah, I get it. So then, regardless of the mechanism, you've seen that, eating X versus not eating X, include Jerusalem artichokes and avocados in your diet, that will change these pathways. You have seen a definite correlation. Different food causes different pathway expression.
Oh, absolutely. Absolutely. And it's not just us. We have literally thousands of literature points, which we also see and, always take into account in our scoring formulation for each of these pathways. We see what kinds of literature support from all of the other scientists that have been working in this area has already shown us. And we incorporate all of those into our algorithms, or, think of them as sort of decision trees or AI algorithms that we've implemented within the Biome system.
Well, it's nice. It's a different way to look at it. Like you said, 99% of the literature out there talks about just species and, from a taxa approach. So you're looking at a pathway approach.
Yeah.
Any sense of how much of a given food or the absence of a food will affect these pathways, and how long it takes, and how much of a change that can occur over time?
Yeah. So this is, you're hitting now on some of the new clinical studies that we're doing. So one of the, actually there's multiple studies that we're currently doing with many partners where we are doing longitudinal analysis of your microbiome. So for example, we can get a sample of your microbiome every week for 10 weeks or 12 weeks and see what are the changes that happens when you follow the diet that we have recommended to you. And this is one of the ongoing studies we have right now. Another study we have is looking at how to capture the inflammation that, or inflammatory activities that changes, especially for people who have chronic inflammatory diseases like IBD I was telling you about earlier, right? So, what happens in IBD, as just to give you a very concrete example, is that, once you start suffering from IBD, it never really goes away, right? So you have to continuously manage it by eating the right foods or taking medication. Unfortunately, the traditional medicine asks you to take a lot of very strong medicines like biologics, and these biologics can have lots of side effects and what have you. But what we are trying to sort of figure out is, can you control and can you manage a low level of inflammation in your gut just by eating the right foods? And in order to do that, some of the studies that we're doing right now are essentially tracking longitudinally, right? Every week, every month, we look at one sample of your gut microbiome, and we say, hey, what has been changing? And then when somebody gets into some kind of a flare up of one of these inflammatory diseases, we say, okay, what just changed in the microbiome that caused them to become suddenly inflamed? And then that inflammation may stay for like a few weeks, and then that may go away. So an inflamed gut can go from healthy to very sick to healthy very, very fast, within a matter of a few weeks. So we need to track all of those things. So we are currently doing these clinical studies to tell exactly the answer to your question about what are the changes in your gut microbiome that are causing the inflammation to go up dramatically. And then the next step after that is for us to say what kinds of foods can, in fact, prevent that inflammation from happening when you look at this microbiome over time.
Well, you'll see change, no doubt. But again, if you see change in the prevalence of a pathway, up or down regulation of it, I guess I could say, then that's fine. But what led to that? And that would probably be the appearance or disappearance of various species.
It's again, it's the food that you eat, right? So your food essentially provides the substrates for the microbiome to activate or deactivate various pathways. So for example, if you eat a lot of fibers, specific kinds of fibers, complex carbohydrates, then many of these microbial pathways start to become active. And if you do not eat those fibers, then you are actually preventing these pathways from becoming active. So the starting point for your microbial pathways to become active is definitely food. And so this is why we believe in Viome that getting the right macronutrients and micronutrients in your food is really the best way to manage your chronic illness. Now, I want to mention two things here, right? So there's when I say food, I'm thinking about complex carbohydrates like prebiotics, right? They're also known as prebiotics that can help your current microbiome or organisms in your microbiome become more active or more abundant in your microbiome. That's for existing microbes or existing genes in your microbiome. Okay. There's also the possibility that if you do not have particular kinds of organisms or genes, microbial genes, we could give you probiotics, right? So there's been a number of studies that show, for example, that Akkermansia is a very beneficial, probiotic for certain kinds of chronic diseases, like including diabetes, by the way. There's a lot of literature that supports that. So there can be probiotics and there can be prebiotics. Those are the two different kinds of things that we look at. And sometimes we can also give you, instead of activating your microbiome to generate certain metabolites, we can, if there's a severe lack of metabolites, we could also give you the actual end result, meaning that metabolite itself, to boost your activity for a short period of time. That's not sustainable because that's not continuously generated by your microbiome. It's just a short-term fix while we actually improve your microbiome so that it can give you the long-term fix, right? So these are all different ways of putting things together. You can give probiotics, you can give prebiotics, you can give metabolites directly, and you can in fact combine all of these things. In fact, that's exactly what we want to do next, right? We want to take the insights of what is active and what is not active in your microbiome, turn that into supplements, right? And specifically for you, right? So it could be like, maybe we discover that there are 9 things that we need to give your microbiome in order to restore all of these different pathways and the metabolites that we see a lack of. and we can combine those 9 ingredients into 3 or 4 pills or, supplement pills that we could give you as a personalized pill pack that, we mail that to your house, and you can, literally, pop it in every morning. And that becomes sort of your personalized sort of wellness sort of regime, right? That's kind of where we're going next.
That's very cool. Would you consider an unprocessed, non-irradiated food to be itself a prebiotic and a probiotic? Because, I would think natural things, like if I pull a banana off a tree and I eat it, it should have its own associated microbiome with it. So absolutely.
So there's a lot of probiotics in natural food that you can get, salads and all kinds of different things, right? So fruits as well, right? And again, there's no such thing as a single food being, good for everybody. We have seen that, broccoli can be good for you and it can be bad for me. On the other hand, the banana can be great for me, but could be terrible for you. It's, one of the things that we published in a paper recently is that, if you and I eat, let's say 2 very well-known foods like a banana and bread, okay, it could be the case that, my blood sugar spikes when I eat bread, but does not spike at all when I eat banana. But your blood sugar spikes when you eat a banana, and it does not spike at all when you eat bread. And that difference between you and me is pretty fundamental for us to understand. It's not like, I can tell you everybody that, banana is a good food because it's good for me, not good for you, right? So it's the same thing for pretty much most foods. It's not like there's a universal great diet for everybody. It has to be personalized. And that's why we think we need to understand your biology first through some kind of home tests, like our gut microbiome test. And based on that, we can give you what will work for you, but definitely, does not definitely work for anybody else.
This is like a whole continuum, I guess an aware person could get like a food sensitivities test, but that's maybe just things to avoid. But you can continue into the health part of the spectrum and, go through Viome, for instance, and see what foods to add or to have more of as well. So you get the more complete picture and a personalized one.
Totally. I think that's a great way to think about it. So it's definitely a continuum, and and that's actually a way for us to also talk about the microbiome and its role in the overall biology, right? Microbiome is a very critical element. It's a key element of your biology, but it's not the whole thing, right? There's a lot of other things that are going on in your body, like for example, how your human genome is being expressed. And how the interaction between your human human gene expression, and the microbial expression of the pathways, right? What the interactions are between the two. And that interaction, in fact, causes the main sort of personal, biological issues or health that you experience. So what we thought in Viome is, of course, that the microbiome is a great place to start, and we've made a lot of progress with that. But we're now at a point where we are saying, let's start completing the picture and start adding the human host part of the puzzle as well. And so, we've been doing R&D on blood transcriptomics kit, which is basically a kit that similar kit as your gut microbiome kit that you get at home, but you just prick your finger. Get a few drops of blood and send it to our lab, where we see what is the expression of genes in your human, blood, and now we see the interaction between the gene expression that's going on in your human cells and the gene expression that's going on in your microbial cells in your gut microbiome, and the two of them together gives us a much more complete picture of what is going on in your body, instead of just looking at the microbiome alone. So that's kind of the next step that we are talking about here. And we have started to do a number of studies. And we've started noticing, for example, that, we can predict, lots of things about your phenotype once we see your gut microbiome and your blood transcriptome both together. So, for example, we can predict your age just by looking at your microbiome and your blood transcriptome. We can say, you're probably within, these, sort of this 8 plus or minus 8 years of your actual age. We can say whether you sleep well or not by looking at your microbiome. We can say whether you took an antibiotic or not. That's probably a little bit more obvious, right? Because antibiotic has such a big influence on your microbiome. We can tell what kind of bowel movements you have, right? We can say, what your general health is. So we can say all of these things just by looking at your data without asking you any further questions, right? So what that's leading us to believe is that once you get this picture, the full picture of your biology, we are going to be able to, create, almost like a digital, a virtual biological model of your body, right? Given all of these different pieces of, the puzzle. And based on that, we can start telling you about, what kinds of functions are active, what kinds of functions are not active. And of course, we can give you some lifestyle advice and food advice and everything else going forward.
So how specific are your recommendations right now? If I send my kid in today, you get my kit, and how specific will the recommendations be? Because the reason why I ask is years ago, I had my microbiome sequenced by a number of companies. I won't say who, but they basically told me nothing. I don't know if they were allowed to, but they said, "Oh, you have this species and that species and this species," and that was it. They couldn't say any more.
Yeah, you should definitely give it a try right now for Biome. The science has come, made a lot of progress, and in our case specifically, what we tell you, so we tell you two things. Okay, there's one section of our app that's called the results, and the other section of our app that's called recommendations. And the results basically tell you what is going on in your microbiome. So it tells you the long list, very long list, in fact, of the species in your microbiome. But more importantly, it tells you the pathways that are active in your microbiome. Okay, so for example, it tells you that your butyrate pathway is highly active, and your LPS pathway is not very active, which is a good thing, by the way, right? And it can tell you that your oxalate pathways are sort of average, right? So there are 20 such pathways that it tells you about regarding your microbiome functions, right? Think of these as functions as opposed to just species, because species are, by themselves, species are just a kind of a curiosity, It's not.
You told me,
Yeah. So these functions give you a lot of insight into what is going on. So that's the results part. The recommendations part are also pretty specific. It tells you, it gives you like a list of hundreds of foods, and it classifies them into your superfoods. So there's, typically, 15 to 25 or 30 superfoods that every individual has. And, these are your go-to foods. And, you could have, for example, broccoli on your superfood list because maybe, that you have some inflammation going on, and maybe there's some liver detox support needed, and broccoli is great for that, so you could have that. But for me, it could be on my avoid list because I have too much, maybe there's thyroid function support that I need, or maybe there's too much sulfite gas being produced and so forth, so maybe avoid for me the same exact food. And then we also have a list of about 20 foods or 30 foods that definitely you should not eat too much of, right? You should be avoiding that, meaning, don't eat more than once per week or per every other week or something like that. And again, these superfoods and avoid foods could be quite different from each individual to individual. So that's the level of specificity we go to.
And then what if I get, my microbiome evaluated by you, and then I do the test again later? What do you recommend? A month later, 6 months later?
We recommend, at this point, 90 days later, 3 months later. So in fact, when we give you our recommendations, the food recommendations, we say these recommendations expire after 90 days and you need to retest. And of course, not everybody retests, but still we know that there's, your microbiome is dynamic, right? It's active, because we are checking for activity. So once you follow the recommendations, it's definitely going to change your microbiome. And at this point in time, we're doing lots of studies to say what is the optimal time to retest, but we think 90 days is a very good time for a retest. And, you can see the the changes in your microbiome if you followed the recommendations. And the changes manifest themselves as scores changes. You'll see differences in your, activities in your microbiome. You'll also see changes in your recommendations themselves, because if you ate a certain set of things, you're definitely going to change the activity levels in your microbiome. So that's going to definitely cause variations in your in your core pathway activities as well.
Okay, very good. What kind of anecdotal comments have you gotten from people that have sent in samples? When they've made a change and then they've come back to you and sampled again. Anything that sticks out at you that's really interesting?
Oh yeah. I mean, people have come back to us and said that, what they used to suffer with rheumatoid arthritis and their, joint pain and so forth has started to vanish. People have lost weight. People have, started feeling, clear in their head, no brain fog, that kind of thing. People have started to sleep better. I mean, there's actually a whole video on our website about, customers of Viome who have come back to us with, these kinds of testimonials. We also are doing systematic studies, right? Not just anecdotes, but we're doing systematic studies where we see the sort of the consistent and, repeatable kind of changes that happen in the microbiome, and therefore in the phenotypes, right? All the symptoms and lifestyle factors that people care about. Those changes are also we are beginning to quantify those changes through a number of these studies, which are going to be many of them are going to be longitudinal, in the sense that we track how well people are following the recommendations and what the symptoms changes are over time. So we are doing those kinds of studies now. And we will start to publish the results from those studies as soon as we have them.
Okay. Well, very good. What's the best way for people to, first of all, find out what services you offer, to get a kit, maybe to read papers that you're putting out? What do you recommend?
Just go to viome.com, and we put all our, first of all, our kits are, $149 at this point in time. And many times there are sales that, bring it down to $129. So it should be, something that everybody should be doing, in my mind. And in our website, there's also a science section. And if you go to the science section, there's, a lot of information about the fundamentals of, foundations on which we build our recommendations. If you're interested in the papers, you can just go to Google and you can type in my name, Guru Banavar, for example, or you can simply say Viome and metatranscriptomics, which is the technology that we use and the science that we use, and you will see our published papers. Some of them are published in peer-reviewed journals. Others are on pre-print servers like bioRxiv, and you'll see all of these things. And obviously, you can respond directly on these websites, on these online sites. And if you do so, we will be happy to, respond to your comments anytime.
All right, that's great. Well, Guru, thank you for all that you do, and I appreciate you coming on the podcast.
Thank you very much, Rich. This was a pleasure.
You're listening to the Future Tech Podcast with Richard Jacobs. Future technologies such as artificial intelligence, stem cells, 3D printing, gene editing, Bitcoin, blockchain, the microbiome, quantum computing, virtual reality, and exploring space are much closer than you might think. In fact, many early versions of these technologies are in play right now, and the companies that are using these technologies are the focus of this podcast. My goal for you, the listener, is to learn from these podcasts. You may very well learn something that may change the course of your life for the better, steer you towards a new career, or give you insight into addressing a thorny medical problem. Remember, this podcast and its content is informational in nature only. No medical, tax, legal, financial, or psychological advice is being given. If you enjoyed the podcast, please listen, subscribe, like, and tell your friends about it. Thank you.